Good evening, everybody. Today we have failed to go live at this particular moment, but we thought we're going to prepare a webinar as is anyway. And I'm quite glad to introduce Maria Dimitriadi, who's working in SMA, looking into worm models. She will elaborate a little bit more. Maria has a lot of experience in the area. She moved to UK, Hertfordshire University from the US. And apart from being a great scientist, she's also a very lovely human being and cares about SMA people quite a lot. So hopefully we will continue our ongoing collaboration for years to come, and we can have a lot of fun with worms and without them as well. So do you want to give us your presentation, and then hopefully at some point I'll get back to you with some questions.
Okay, lovely. Thank you so much, Gennady, for the introduction. I'm just going to share my screen now, and hopefully it's going to work. Let me just try to make this into a slideshow. Oops, nope. Let's go back. Okay. Oops. I'm trying to move the icon so I don't see my face directly. Okay, so thank you so much for giving me the opportunity to be present here today. And it's a great pleasure to share with you our work on spinal muscular atrophy. So as Gennady kindly said in the beginning, my name is Maria Dimitriadi. I'm a reader in molecular genetics here at the University of Hertfordshire. And I hope you wouldn't mind me today sharing with you how we can actually use a small roundworm with a scientific name, Synarchdiotis elegans or C. elegans, which actually shares many biological characteristics with humans in order to understand diseases like spinal muscular atrophy. So what I'm going to be doing, I'm going to be focusing on the first part of my talk on introducing you to the roundworm C. elegans, because I would like to make everyone really excited about the worm model, as we C. elegans scientists are. And then I'm going to switch gears and talk to you about spinal muscular atrophy and what actually lessons have we learned from a tiny worm for SMA. So in my lab, we use C. elegans, which is a 1 millimeter roundworm, to study spinal muscular atrophy. And now I'm going to show you a video of C. elegans and how it looks under the microscope. So C. elegans, as you can see, and you'll probably appreciate from that video, and I'm going to play it again, is actually 1 millimeter roundworm. And what is fascinating about it is that it's transparent. So if I try to play the video again and I slowed it down, so what you can see here, this is the head of the animal. These are the non-fertilized eggs. Down here, you see that the eggs have been already fertilized because the animal makes its own sperm and egg. This is the vulva, so eggs will be released in the extracellular environment. And at the end, you are going to see this is the intestine, and this is the tail of the animal. So apart from it being that it's been transparent and it's great for us, for C. elegans scientists, is also that because it's been transparent, we can actually track its development under the microscope. And apart from this, we can actually amend its DNA or its environment, and we can see how that it will affect its behavior. The other great thing about C. elegans is that it has a very short lifespan, so it only lives by 3 weeks. Its muscles and neurons are very similar to the ones that we have. And most importantly, 40% of the genes that can cause a disease in humans have exactly the same gene in C. elegans. So one of the things I always say to my students here at the university is that C. elegans research has contributed not 1, not 2, but actually 3 Nobel Prizes to the way we understand science today. And these were given back in 2002, 6, and 8. But I just want to give you a brief overview, and that will be very brief, on how actually everything started. So Sidney Brenner is actually what we call the father of C. elegans. So he realized very early, by in the 1960s, how the powerful genetic tools of C. elegans. And he's a South African biologist that moved to Cambridge, and he was the first one to look at the DNA structure when it was firstly discovered by Watson and Crick. So he actually demonstrated that there were specific gene mutations that can be introduced in the DNA of C. elegans. And he identified more than 600 mutations in the worm model. And these mutations were affecting its development, but a large number of these genes were affecting the nervous system. So it's very important to understand here that if you've got a mutation, this mutation is going to affect our phenotype, is basically what we see. But by affecting the phenotype, we'll be able to understand what is the function of the gene that is being mutated. So this is something that Sidney Brenner was working on. So along with him in 2002, it was Robert Horvitz and Salston that received the Nobel Prize, and they all three use C. elegans. And I just want to briefly mention here that Horvitz was the first person that actually used C. elegans and discovered the first 2 genes that can cause death in a cell. So this process we now know it's called apoptosis, or programmed cell death. And in C. elegans specifically, we know that the adult animal will have 959 cells. But Horvitz realized that there were 131 cells that were dying at a specific point all the time in every single animal, like as if this was completely programmed. And that's one of the reasons why he joined the team for his discoveries on programmed cell death. And at Salston, as you can see on the right-hand side, is a scientist who actually, what he did is he sat under the microscope and he tried to trace all these 959 cells that we know nowadays they exist in C. elegans. And if you, what he specifically said was that imagine that you're watching a plate that's full of grapes, and you're focusing your eye on one specific grape and trying to see how that will move when all the other 958 grapes move at the same time. So imagine how difficult his job is, but now you can understand why people win the Nobel Prize nowadays. So basically, Salston, what he's given us now is a complete invariable, as we call it, cell lineage. That means as a scientist, I know exactly where every cell is. I know exactly its position. So I can use very interesting microscopes to ablate and kill a specific neuron, for example, or a specific other type of cell, and can find out how ablating, this cell affects the phenotype of the worm. So moving forward, and I'm not going to spend a lot of time on Nobel Prizes, but I just want to briefly mention here that in 2006, Andrew Farr and Craig Mellon used C. elegans, and they received the Nobel Prize because they identified RNA interference. And basically this is a process that we use a lot nowadays to stop the function of specific genes so that we can study them. And all the work was done in the worm. And lastly, back in 2008, and this is the time when I start working on SMA and starting to learn the C. elegans SMA model. So Martin Chalfie, that you can see in the middle here, is a C. elegans scientist, together with the other 2 scientists that I mentioned here. He specifically used C. elegans, and why is because these people actually got the Nobel Prize because they discovered the green fluorescent protein, or GFP. So this is basically a protein that was originally isolated from jellyfish. And how can it describe this better is, for example, think that you're interested to identify how a protein works in a cell. And the other great thing about C. elegans that we said is that it's being transparent. So if I'm interested, for example, on SMN protein, I can tag it with something that will give it a color, like a green color, and I will be able to track this across the animal and see exactly on which tissue it's going to be expressed and where it's going to be important. So what you can see here on the right hand side is basically C. elegans, and as you remember, it's transparent. So scientists, what they've done here is they express GFP tag protein, the green fluorescent protein, in all the neurons of the animal. So you can see how beautiful the animal looks. So moving forward, I need to mention that C. elegans is actually not parasitic, non-hazardous, non-infectious, not pathogenic. So I can give you a plate of worms, you can take them at home and it would be fine. These animals, they only eat E. coli. They're actually free-living nematodes, or roundworms as we call them. They live in the soil across many different temperature regions of the world. And they will survive by just feeding on microorganisms. And on the right-hand side, you see Science, which is one of the very well-known journals in science. And you can see the C. elegans at the front page because it was published when its sequence, when we exactly knew all the letters on its DNA. Yeah, that was back in 1998. So why am I interested in C. elegans, and why there are so many other scientists that work with C. elegans? First of all, it has a very small size. As I said, the adult animal is only 1 millimeter long. It's multicellular and it's very simple. It's very cheap to maintain in the laboratory. So for example, a plate of animals, when we buy it from America, from the consortium there, it's only £7. And actually these animals are shipped by Royal Mail because, as we said, they're non-hazardous, they're fine. They have a very rapid life cycle. So from an egg to an adult, it can take up to 2.5 days, depending on the temperature. Again, talking about what Salson found out, it has an invariant lineage. So I've got a book and I know exactly where every cell is, and it's going to be exactly the same position in every animal I'm looking at. It's transparent, as you already know by now. With C. elegans, we have excellent resources online. So its genome is very well studied. So we know where the genes are, the positions. We know what studies have been done. So it's very, very well annotated database. It's very easily amenable for genetic techniques. So for example, if I have a plate of worms that have a specific mutation and I want to create another mutation in this worm, it will probably take me around 3 weeks to produce an animal that will have 2 mutations at the same time. So you can understand in 3 weeks' time, I will be able to know if what I'm looking at is actually exciting or not. I don't have to wait years to do that. Lastly, we also know a great deal about the nervous system. So as I said, their muscles and neurons are very similar to the ones that we have. So all the mechanisms are highly conserved from the worm to the human. And more than 40% of human genes that can cause a human disease have exactly the same gene in C. elegans. So that's why nowadays there are C. elegans models, for example, of Parkinson's disease, Alzheimer's disease, amyotrophic lateral sclerosis, and so on. So it's been highly used to model human diseases. So what is C. elegans really good for? It's good for developmental biology, for cell biology, if you want to study host pathogen interactions, for neurobiology, which is something that we're particularly interested in when we're looking for SMA, for genetics, aging studies, metabolism, and in general about human health and disease. So if you come and visit the lab here at the University of Hertfordshire, you are gonna realize that we are actually growing these animals on plates. So this is a plate that you see here on the right-hand side, and at the center of that plate, you can see there is a spot of bacteria E. coli food, and this is what they eat. So these animals are very easy to maintain. So you can put a single animal on a plate, and because it self-fertilizes itself, one animal can give up to 300 babies. Another fascinating thing about C. elegans, and this is something I'll use a lot for my students here, is that it can be frozen and it can become alive again after decades. And so if you're not interested anymore in this specific project, you can freeze the worms down and you can make them live again when you want to. So this is one of the ways how I shipped my worms from America to the UK. They were shipped on dry ice, for example, using FedEx. They're very small size. They only occupy a little space. They can be grown in different temperatures, 15, 20, 25, depending how fast you want to grow them. And another interesting thing is, you probably realize, as I said, in these plates, we're only adding E. coli and the nutrients they want. But for example, if you don't use sterile techniques, you might contaminate the plates. So if you open the plate and you start coughing, that's all actually great. So if you contaminate your plate, you'll have to use, we just use bleach solution, the one that we use at home. And the animals that are not clean anymore, they die. However, the eggs are resistant to the bleach preparation solution, and then we can transfer the clean eggs to a plate, and that's how we can propagate the strain again. But also we can grow C. elegans in liquids in large quantities, and this is something very exciting for us, because if we're specifically looking for drug studies, we can grow a large amount of animals in liquid. And you can use different machines. And if you remember, I mentioned before that you can have C. elegans expressing the green fluorescent protein in different tissues. So you can have worm sorters, as we call them, and they can sort GFP from non-GFP looking worms. So you can carry on your experiments. So the worms will go through the machine, they will come out, they will be still live and happy. You will carry on doing your work. So a couple of, a few more points. There are two forms in C. elegans. There is at the top, you can see the hermaphrodite, and at the bottom, you're gonna see the male worm. And I'm not gonna go through in detail its anatomy, but what you probably realize is that the big difference is on the tail. So if you try to distinguish a hermaphrodite at the top or a male at the bottom, you're gonna be looking for the tail structure to be able to distinguish these two. The C. elegans life cycle is very simple. As I said, from an egg here to the adult that you see at the top, it can take 2.5 days. So when the egg hatches, you're gonna have little babies called larvae 1, then larvae 2, larvae 3, larvae 4. This is the time where they start to make sperm. And after therefore, they start to make their own eggs, and then will become adults. If conditions are not great, they will try to preserve themselves, and they form this dower stage that we call here. And when conditions become happy again, for example, if you've forgotten to feed them, they're not gonna be happy. So they will form this dower stage. And when you feed them back again, they will go back and start becoming L4s and adults. So these are just a couple of images so that you know how they actually look in real life. So you can see the eggs here, the L1s, they're really tiny. We don't really want to touch them because they're very small and petite and very fragile. The L2, L3, L4s are up here, a young adult animal, an adult animal. So you can see the intestine here is much more pigmented, and an adult male, and you can see the difference in the tail here. And this is if you zoom with your microscope, again, you can see the animals, the worms. The traces that you see is because they have this characteristic sinusoidal movement, and that's the traces that live on the food. And you can see the little eggs here. And this is something that you're gonna see if you look under the microscope. Okay, so now so I've already told you a couple of things about C. elegans, which I thought they were important so that you can understand what we work on. But now, as I said, Brenner initially identified lots of mutations, more than 600 mutations. And these mutations were causing specific phenotypes. So phenotype is basically how something looks. So on the left-hand side here, you can see the characteristic sinusoidal movement of the worm. And this is what we call wild type or a healthy worm. Now, if you cause specific mutations or different types of mutations, the worms might change their phenotype. And for example, this is what we call a roller phenotype. And these animals basically will go and roll around. They will just gonna follow their tail all the time. And that's what we call the roller phenotype. And this can be caused by different types of mutations. Now, on the left-hand side, at the bottom, you can see another type of phenotype, which we call it the dumpy worm. These worms are much smaller in size, and they're much more fat than a normal wild type animal. Again, this is another characteristic phenotype. And this is what we call the multivalva phenotype. So you might remember at the beginning, I showed you a video of a C. elegans, and I slowed it down and I showed you the area where the eggs are gonna be released in the extracellular environment. So these actually worms have lots of these structures here, but these are not functioning. But this is what we call a multivalva phenotype. And another interesting phenotype is on the right-hand side here, which is called the uncoordinated phenotype. So these animals actually do not really move really well. They're actually gonna move more like this type of behavior. So why this is very interesting to us is because if we introduce a mutation and we don't see a phenotype, then we won't know whether what we're doing to what we are introducing is actually exciting or not. But if we change the phenotype, it's very important for us to try to find out, using the great genetic tools of C. elegans, how we can make these phenotypes become normal again. So before moving forward and now start switching gears, talking about worms and SMA, just want to play a game with you. So I'm going to be showing you different videos, and from what I said, and if you actually listen to my talk up to now, I would like to find out if you can guess what type of phenotype you can see. So let's try with something I think might be more easy. So if you look at the left bottom side, I can show you a video. So you can start thinking, according to what Maria said, what kind of phenotype is this? And you probably think that's a roller phenotype, and you are correct, because as you can see, the worm starts to move around itself. So let's go and look another, the middle one here, and see. So you're probably thinking that worm doesn't really move, as the previous video that Maria showed. And it looks much shorter, as you probably realize, and it looks a bit more fat. So yes, you're right, it's a dumpy worm. Okay, so now let's go and try and look at the top left one. Obviously, these videos have been, have speeded up, so it's not that they'll move so fast. But you can see a little worm and a bigger worm. You probably realize by now this is actually a healthy looking worm. This is what we call a wild type, C. elegans. It's a geneticist, really. I'm gonna show it one more time. Oops, sorry. So if you go to the right-hand side, the top corner, I would like you to focus on the tail. And I'm gonna play the video again. So you can see the tail looks a bit different than the healthy looking worm that you were looking at the previous video. And actually this is a male. And at the bottom of this screen, so now I'm showing you a video of an animal that's actually struggled to move. As you can see, it doesn't have this characteristic sinusoidal movement. So if you thought it's a dumpy worm, you are correct. Okay, so now, so this is very important for us. This is very important for us to be able to identify phenotypes and how we can reverse these phenotypes to make the animals look healthy again. So you probably realize by now that C. elegans scientists like myself are very passionate about what we do and using the worm model. So everything we do has the worm word inside. So we've got a worm book that you can actually go online, and you can read lots of different chapters written by C. elegans scientists in the field. We've got the worm base, and the worm base, again, we can find lots of information when it comes to genetics. Where is it, for example, SMN1 gene identified in C. elegans, which chromosome, what's the position, what studies have we identified up to now. We have a worm atlas. So again, you can go online, you can find out specific tissues, how they look, 3D structures. So it's been very, very highly annotated, all these different websites. And we also have the Worm Show. So the Worm Show is a show that takes place every 2 years when all the C. elegans scientists meet in America. And what we do is there are normally 2 principal investigators, 2 professors that we run a show for an hour. And you probably laugh if you've worked with C. elegans before. So normally I use videos from the Worm show every year to show them to my students here after they had a couple of lectures in C. elegans, so that I can see whether they understand the jokes. And it's always nice to see their faces when they start to smile. And that, you know, that makes me really happy. So what do scientists really do with C. elegans? So there are different types, different C. elegans scientists. You got scientists that use C. elegans to understand development or organ development. There are different scientists that use C. elegans to understand the behavior and the function. Because you remember, we are looking at mutations that affect the phenotype. The phenotype, of course, will affect the behavior. And then we're going to be able to understand more about the function of the gene that actually will cause the mutation. And there are scientists like myself who are interested in using disease models, using the model organism here, C. elegans, and how we can actually mimic a human disease in a tiny worm, and what kind of lessons can we actually learn. So now I'm going to be switching gears, and now I'm going to be focusing more on spinal muscular atrophy and what lessons have we learned from a tiny worm. So you probably are aware up to now that there are different types of genetic models to understand SMA disease pathogenesis. There are mouse models on SMA, there are zebrafish models on SMA. There is the model of the fruit fly, or Drosophila as we call it, on spinal muscular atrophy. And of course, we have the C. elegans SMA model that we're going to be focusing more on today. Again, if I ask you, why do you want to study SMA in C. elegans? You're probably gonna tell me by now that yes, we want to study, for example, because it has a short generation cycle and lifespan. Yes, so they only live for 3 weeks. From an egg to an adult, we said 2.5 days. We know a great deal about their nervous system. So their muscles and neurons are highly conserved between the C. elegans, the worms, and humans. The pathways, highly conserved as well, the presynaptic and postsynaptic. And of course, as we mentioned a couple of times today, more than 40% of genes that cause a disease in humans have exactly the same gene in C. elegans. And that's what we call a C. elegans ortholog. So let me introduce you now to the C. elegans SMA model. So at the top, you're going to see what we already set up to now. This is a healthy wild type looking worm. At the bottom here, so what has happened is we have inactivated SMN1 gene. And I have to mention here, worms, flies, like in mouse models as well, they do not have SMN2 gene. They only have SMN1. So when you inactivate SMN1 in the C. elegans animal, instead of getting a healthy worm looking like this is actually an age-matched control. So what you can see that these SMA worms look, as you would appreciate, much smaller in size. They don't have the characteristic intestinal pigmentation. And they are sterile. They actually won't go, they don't lay eggs because they will never go into adulthood. And they will die at the sort of larva stage. So they're when they're much younger. And apart from this, why we're very interested in this model is that SMA worms will show neuromuscular defects. So the way they move is going to be affecting. And of course, you know that if you want to move, you need to use your muscles and your neurons. So these worms actually are not happy at all, in the sense of they don't move really well, and they do not eat really well. And what specifically we're trying to do here is, what kind of genetic tools can we use, and how can we make these worms happy again? All right, so what I'm going to do is I'm going to show you, so if you want to model something in C. elegans, it has to be very relevant to the human disease. So if I tell you, for example, you know, the C. elegans worms, the SMA C. elegans worms look much shorter, you might think that's not very exciting, Maria, because I'm interested in the muscles and neurons. So what I'm doing here is I need to find phenotypes that will specifically be related to a neuromuscular phenotype. Yeah, so because this is going to be very similar to humans as well. So what happens, as you can see on the left-hand side, this is the head of the animal. And this is what we call the pharynx. And this is a healthy looking worm. So I'm going to show you a video now. And so now what happens in C. elegans is that the worms will feed on bacteria, microorganisms by using this small and discrete subset of muscles and neurons that's found in the head of the animal. And so you can see now the video. So this is the head of C. elegans. And you can see this structure here, that's what we call the grinder, that is going to move back and forwards while the animal is present on foot. So when they eat E. coli, they will spit water out and so on. And if you try to slow down this video and count how many times does this grinder move back and forwards, you're going to realize that if you count this for 1 minute, you're going to count 200 and 250 beats or pumps, as we call it, per minute. On the other hand, now let me show you what happens when you inactivate the SMN1 gene when it comes to what we call this is called the pharyngeal pumping assay that's controlled by muscles and neurons. So look at that, what's happening in the SMA worm. Again, this is the pharynx. You can see the grinder is moving, but it's moving a bit differently it has a bit more arm coordinated phenotype. And if I ask you, and actually you don't even have to slow down the video. If I ask you to count how many times does it move, you probably be able to do this without slowing down the video. And why this is because these animals, they will only pump at 30 to 50 beats per minute. Whereas remember, the healthy worm was pumping at 200 or 250 pumps per minute. So they have a dramatic neuromuscular phenotype that's been immensely affected by the loss, the depletion of SMN. So this is just an example of how C. elegans scientists try to take advantage, as we call it, of a phenotype that's been caused by a specific mutation. Here, specifically, we're talking about the SMN mutation, and how we can use this phenotype to identify cellular molecular pathways that are important for this disorder. So, so we've used in the past, for example, the pharyngeal pumping assay and lots of other assays, which I'm not going to have the time to discuss with you thoroughly today, but I'm just going to tell you a story. And we initiated genetic screens. And what this means, genetic screens, is that if I inactivate a gene that causes a specific phenotype, I've got the advantage of using C. elegans, and I can inactivate, for example, 20,000 genes one by one if I have a very nice automated readout, and can find out which is this gene that when I inactivate it, it will make my worms happy again. All right, so this is what we did back, for example, in 2010, and I'm gonna explain you later on why I'm going back a couple of years, many years ago, because we were looking, for example, for genetic modifiers. And you probably know that the SMN2 gene is a genetic modifier. So what a genetic modifier means, this is a gene that can modify another gene. So SMN2, if you increase levels of SMN2, then the phenotype gets better. So SMN2 is supposed to be a genetic modifier. So that's what we're trying to do. We try to identify, using the worm, because we can get a very quick readout, genetic modifiers of SMN. So which are these genes that when we alter them, it will make our worms happy again? And we can do very, very, very quickly in C. elegans. So when we did, what we did back in 2010, we had the C. elegans spinal muscular atrophy model, and we had collaborators working on the fly model, on Drosophila spinal muscular atrophy model. So we identified genes that we found that they were making our SMA worm model happier or even worse. And then we went back to our Drosophila fly collaborators and asked them the same question. Can you find out if the same gene in the Drosophila, in the fly model SMA, whether it makes the flies similarly happier or worse? So, and why I'm saying this, because I just want to stress out here that when you find a mechanism that's conserved, for example, from flies to worms, it's more likely to be important for human function as well. So I'm going to share the story with you today. So when we did this, we identified lots of different genes. And lots of these genes were working, for example, on RNA processing pathways, which is something that's clearly associated with SMN function. And I'm not going to be focusing on this. But we also identified that our genetic screens, and these are completely unbiased screens, they were pinpointing to a pathway that we know is called endocytosis. And basic endocytosis is a pathway where molecules used to enter a cell. And this was something back in 2010 that was never talked about before on SMA. So I'm not going to go through all the different experiments we did, but what I would like to point here is that when we start talking about endocytosis, we thought, for example, endocytosis is highly used by neurons. And specifically, we call it a neuron synaptic endocytosis. So what you see here is basically a neuron. And here at the bottom, you're going to see the muscle. So in the neurons, obviously, because these are high energy demanding cells, you have to have synaptic endocytosis happening and things that have to be recycled in a very fast mode. So this circular compartment here is what we call a synaptic vesicle. It's a vesicle that will carry, for example, a neurotransmitter. And neurotransmitters are basically chemical molecules that help, or messengers that need to pass a signal from the neuron to the muscle and to other cells. So this vesicle basically, you can see it has a specific cycle. So when it reaches here, you're gonna be filled up with what we call the chemical signal, the neurotransmitter. The neurotransmitter is gonna be traveling down to the membrane of that neuron cell. You're gonna have a couple of different events that take place, like docking, priming, increase in ultracellular calcium. The vesicle is going to fuse with a membrane. You're going to release the chemical signal, the neurotransmitter, and the neurotransmitter then is going to pass that signal, for example, to the muscle for muscle contraction and so on. So we were thinking whether there's a problem, why endocytosis was coming up in our genetic screen, whether there was a problem with synaptic endocytosis in the neurons. So the first thing we wanted to find out is whether in the C. elegans SMA model, as I told you previously, in the area here, you've got lots of proteins and lots of molecules that take part. So we want to find out if we reduce the SMN levels in the worm, whether the proteins that are important for this endocytosis are not going to be localized properly. Okay, so this is the first question we wanted to answer. So we looked at a number of different proteins to find out whether these proteins are specifically for endocytosis can be affected when SMN levels drop. And this is exactly what we did, but we don't have to go through in detail how we've done this, but which we measured basically the abundance and distribution of these proteins, because we'll be able to tag them with a GFP protein fluorescent tag that I was telling you before. And then the other important thing is, if so if we, for example, we can see that there are defects in these proteins, how do these motor neurons actually look in a high fluorescent sort of, in an electron microscope, yeah, that uses, so we can see great details in the motor neuron. So we want to find out whether by looking at the motor neuron in the worm model, we can see endocytic defects, whether, for example, these vesicles that I was showing you before, they are not localized properly. So as to cut a long story short, yes, we've done all these experiments in the worm, in the C. elegans SMA model, and we found out when the levels of SMN drop in the C. elegans animal, then the presynaptic structure of these proteins actually is being highly affected. So that was very exciting for us, because this is something that was originally suggested by unbiased genetic screens. So just to give you a bit of cartoon, so this is basically the motor neuron, the neuron, and that's the muscle. And here you've got lots of different vesicles. And if this is what happens in the wild type, in the healthy worm, and this is here what's happening in the SMA model. And the SMA model is called OK355. This is how the mutation is called, but you don't need to remember all these details. So basically, if you look at the purple things, these are vesicles that carry, for example, these are called dense core vesicles. They carry different types of molecules, but you see the number is the same. We didn't see any difference. If you look at the green structures, these are the synaptic vesicles that I was showing you before. And actually we find out the synaptic vesicles that are used through endocytosis are much fewer inside, much fewer in the number, and they don't localize properly. And also the red structures here, these are the proteins I was telling you before that they help endocytosis, synaptic endocytosis to take place in the neuron cell. And you can see that these structures actually, they're much smaller in size, but that you've got more. So you can clearly see that they are, when you reduce the SMN levels in the worm model, endocytosis in the neuron is highly affected. And I forgot to mention here that we also saw these huge structures in the neuronal cells of the C. elegans SMA model, but not in the healthy ones. And these structures, we call them cisternae. And that means these are characteristic cellular structures that are formed when there's a big problem with endocytosis. So we're very excited about this study, because something that we found back in 2010, moving forward with more molecular and cellular approaches, we try to pinpoint actually why is the worm leading us to endocytosis. And just to summarize it, just to add one more question here, you're all aware that SMA patients, apart from the neuromuscular defects, they might show some other type of defects in other tissues, like for example, the heart, the intestine, kidney, and so on. So we were interested to find out whether the defects that we see in endocytosis are also found in other tissues, because that means our model is probably might be mimicking the human disease in a very well pattern. Just to cut a long story short, again here, I'm not going to go through the details, but I want to tell you, we can very easily study endocytosis in C. elegans. So worms have these green cells that you can see here, 1, 2, 3, 4, 5, 6. We call them coelomocytes. These cells, what they do is they're important for endocytosis, basically. And again, we can use the genetics tools in C. elegans. We can make them green by, again, expressing the G fluorescent protein, the green fluorescent protein in the coelomocytes. So what we're looking at is how many green cells can I see when the SMN levels drop? And you can clearly see on this chart here that when SMN levels drop, the number of GFP positive cells was dramatically reduced, telling me that coelomocytes that are important for endocytosis are also affected when SMN levels drop. And then another interesting thing that we found is in the hypoderm. So when we were looking at the electron microscopy slides, I remember we were looking at these structures here, which I didn't know that time what these were. And I was trying to find out what is this structure that in my healthy worm has all these nice, circular structures, but in my SMA worm, it does not. And obviously I looked back in the worm atlas and we found out that these structures here are called multivesicular bodies. And the multivesicular bodies are basically structures, which we call them late endosomes, and they're important for endocytosis again. And this was fascinating because, again, we identified another interesting characteristic that pinpoints to the fact that when SMN levels drop, endocytosis is highly perturbed in the C. elegans SMA model. So this is just a very quick summary of, this is almost like 5 years of work. But what I would like to stress here is we started genetic screens that were completely unbiased in C. elegans and in the Drosophila in the fly model. We identified 50 genes that were modifying both phenotypes, both in the worm and in the fly. These genes were pinpointing us to 2 pathways, and endocytosis was a pathway that was not previously implicated in SMA pathogenesis. That's why I wanted to move with this forward. Again, we follow a series of experiments, and then we ended up identifying that depletion of SMN caused endocytic defects. And everything was done in invertebrates, like it was done the initial work was done in C. elegans and Drosophila, and then everything else was done in the C. elegans SMA model. And why this is fascinating to me is because this is something we identified the genetic screen back in 2010. That back in 2010, a tiny worm told us to look for endocytosis, for example. Then in 2016, we published our work, but then at the same time, with our collaborators also, in the Wirth lab in Germany. You probably have come across these papers before, but it was fascinating to me to see as a C. elegans scientist that their modifier genes, for example, NCALT, neurocancyl delta, which is involved in endocytosis, and when the levels of that gene, of that protein drop, it protects humans from getting SMA. So to me, it was, again, exciting to see how something that was said by the worms back in 2010, it was followed up by C. elegans scientists. And then independently, the Wirth lab identified a modifier gene called NCALT. And later on, they also published at the same time more work on plastic and coroning. And they pinpointed again endocytosis that seems to be important for SMA pathogenesis. So having said that, I like to stop here. And I just wanted to tell you that we've learned exciting lessons from a tiny worm. And I would like specifically to thank the lab and all my collaborators across the years. I'm very happy, and thank you so much for the invitation today. And don't forget that tiny worms actually can tell us important things about a human disease like SMA, something I've been working on for many, many years now. Thank you. I'll be happy to take your questions.
Well, thank you. That was very fascinating. I really enjoyed listening and learning about your work. I mean, I've come across it anyway, but it's also good to hear from somebody who done all of this, you know, some explanations. The way I think the way I understand things is basically when you work with worms, the question I think, let's start with first question. Do they feel any pain?
This is a good question. So they don't really feel pain. They have receptors that, so there are studies, there are labs that actually can try to sort of understand pathways that are relevant for pain regulation and so on. But yes, they do not feel pain. So this is something that at least works really well for us, for C. elegans scientists, because we can also minimize, you know, the suffering of the animals, or of the suffering of, you know, if you use a C. elegans, for example, to do a screen or a drug screen, you don't have to sacrifice other mammalian models. So C. elegans is one of the organisms that is supposed to be able to, you know, replace and, you know, in the future reduce the amount of animal models used in understanding a human disease.
Well, that's exactly, I think, the point I was trying to get to, because obviously killing 5,000 mice for sake of argument, comparing to this is, you know, for the animal trials, well, this sounds like a very good model. So that's one side of it.
Yeah,
but also,
sorry, Jeanette.
No, go.
No, and this is something that I would like to stress here, that many mammalian collaborators will come to us for a quick readout in a C. elegans SMA model, for example, because we can test things very quickly, especially and very efficiently, especially we have got a high throughput technique. So imagine that you can have a plate that has 96 holes, as we call a 96 well plate, and you can put a single drag in each of these wells, and you can put a single worm, and maybe the worm has a phenotype, because worms actually swim in liquid. That's another neuromuscular assay that we use. So if the worm doesn't, has a phenotype that doesn't swim well, but you can immediately have a quick readout and see which of these 96 drugs can make the worm swim better. So hopefully
Okay.
I would, this is
Interesting. I also noticed no females.
yes, they do not exist. So we have hermaphrodites, which are largely found, and they make their own sperm and egg. So actually they don't any males, but males only exist sort of 0.1%. So if you start digging in the soil, there's no way you're gonna find very easily a male one, but we can actually change the genetics, so we can heat shock the animals, and because there's going to be a change that will happen in the chromosome, then the hermaphrodites will be able to throw babies that will be males. So this is how actually we can maintain males in the lab. We can cause specific mutations, and we use males actually because we want to be able to cross animals and generate like a double mutant animal, an animal that will have 2 mutations. So you go a hermaphrodite that will have mutation A, say for example, SMN mutation. Another animal that you're interested in has another mutation. One is the hermaphrodite, the other is male. You cross them. The male sperm is stronger than the hermaphrodite sperm. So that's why a hermaphrodite by itself can give rise to 300 babies, but if a male sperm is used, it can give rise to 1,000 babies.
I mean, 300 babies, I don't know what to do with one. 300 of, well, I think it's way beyond my means of support. Other interesting thing I've spotted. So you were talking about modifiers, the SMA modifiers. You said, didn't you say 15 different genes that can, or say identify 15 genes that modify SMN defects?
Yeah.
Are these 15 different genes translatable to humans as well? So it's not, what I'm saying is these modifiers you observe in the C. elegans, are they present in humans?
Yes, not all of them. So so normally when you do genetic screen in C. elegans, our screen only identify 15 genes. So we are lucky and unlucky at the same time, in the sense of the list was really small. But there are other screens that might give you 500 genes. And then probably you start thinking, which gene should I be looking at? But the great thing is that these genes were pinpointing to RNA processing pathways, which is clearly associated with SMN function, but they're also pointing to endocytosis. And they might not be pointing directly to the endocytosis, but maybe that one out of the 15 genes might not have a direct role in endocytosis, but was acting and working together with another gene protein downstream that it works in endocytosis. So when we did the interaction map afterwards at the end, we looked at all these genes and what genes they interact with in the pathway. And this is when the unbiased genetic screening C. elegans told us, try to look for endocytosis. You might find something interesting there. So yes, not all the genes have orthologs. Yeah, so, and as I said, 40% of genes that cause a human disease have exactly the same gene in C. elegans. So it depends, but at least for SMA, we're very lucky because we have the SMN1 ortholog there.
It's, again, I don't know whether this is a naive question or not, but I understand that we can knock out the genes from models. Yes. Can we, for the lack of a better word, can we knock them in?
Yes, yes, of course. We can, because it's something that we did. So all these phenotypes that you see in C. elegans, you have to prove to, when you want to publish, or to the other researchers, you want to prove that that phenotype that I'm seeing is because the fact that I've inactivated SMN, not because something else was inactivated simultaneously and I never found out. So you have to rescue. So we have to put so when we put the wild type copy of SMN in the C. elegans SMA model, we saw that we can see a rescue of the phenotypes that we see. So yes, you can put, and now with CRISPR, obviously you can do many more things faster and more quickly, as we used to do when I was a postdoc back in America.
Okay. Moni, do you, I mean, do we, because we're not really live, we don't really have many questions just
Yeah.
yet. So it was very fascinating talk. I'm sure there are some people will be coming back to us asking a bit more questions, but I think since we started a little bit late, I'll allow you to go home to your children.
Thank you.
They're missing their mommy, or probably playing computer games with their daddy.
Or they're sleeping already.
Oh, they're sleeping already.
Hopefully.
Well,
Hopefully.
you're lucky. My one is looking at me with her beautiful eyes and smiling. So
Oh,
I'm
bless
going to be
her.
doing bed routine soon. Moni, who we have next?
We have physio with Marion on Sunday. We have currently got a poll up in the TreatSMA groups as to what talk people want. So we can either do equipment and SMA part 2 or returning back to school. So if you haven't already, people need to head over and vote so Marion knows what she is going to be covering.
Okay. All right. Well, Maria, thank you for this talk. It was, I thoroughly enjoyed it.
Thank you so much. I hope the language was quite late and I didn't use many, you know, high
we all need to
terminology.
learn. We all need to learn.
I've got no like background in this kind of thing at all. I do art and textiles and fashion, and I understood it like my mind is blown, but I understood it, and it's really interesting.
Oh, thank you so much. That's the nicest thing I've heard today. And I do hope things are going to get better. So I really would like to, feel free to come in the lab and look at the worms, see what we do. And we really want to help, the charity and, organize events with, the members there. And this is something that I'm really looking forward to working with you guys in the future. So please don't forget about me.
No, we won't.
Definitely not.
Once again, thank you for today. I would like to reassure everybody that no mice have been harmed in the process of making this video. Not so sure about the worms, but knowing Maria as I do, she does keep her worms very happy, unless she specifically looks into making them SMA related. And even then, she still looks after them pretty well. So nobody panic. It's all good, in the good hands. Well, I wish you good night. Well, we'll be inviting you at some point soon.
Yes, I'm going to stop share so I can see your face properly.
Okay. All right.